Good evening, Dr. Dan Guerra here from Authentic Biochemistry Studios in the inland Pacific Northwest. At least that's what it is outside that window right there. I'm doing part 2 of Cancer and Aging Immunosenescence, as you can see by the title. As I said, it's Authentic Biochemistry, and it is, as you can see, a rare video podcast presentation. Today is indeed the first day of summer, the 21st of June, 2021. A lot of 20s and 21s in that date signature, right? At least 2 21s and 1 20, in fact. All right, so let's get started with all of that numerology behind us. All right, so let's see here. Slideshow, that always works. And we're going to start with the current slide. Okay, and we're going to hide that. Okay. So let's get right to this. Sorry, I always make that mistake the first time. All right, so we were deep into discussing the stress response, so I'm not going to give you a huge reiteration of what we talked about just a few days ago. Let me explain the stress response, termed cellular senescence in aging, links multiple pathologies of that process, both degenerative and what I call hyperplastic, which means hyperplasia as the noun, and that would mean, of course, the potential for tumorigenesis. Cellular senescence is unlikely to explain all the aging phenotypes. We've gone through this in great detail for the last 6 months. Hopefully that I'm confident that you will agree with me on that. In other words, we can look at cellular senescence as a process where basically cells stop dividing and we get a couple of specific phenotypes, such as the secretory phenotype. And we know that there's an associated autophagy and apoptosis, sometimes necrotosis. And we know that there are changes in gene transcription because of transcription factor expression or lack thereof in different cell lineages. And we also know there's DNA damage and DNA repair mechanisms. But all those processes I just talked about aren't all confined within cellular senescence. So obviously there's some kind of harmonization of multiple processes that otherwise do not promote aging, at least not the kind of rapid high morbidity aging that we reach towards the end of the life cycle, right? So it's not just the rate that's changing for the aging process, and I'm not even sure I would argue for that. It's more that it becomes tangibly a unique aspect of cellular fate. That is, it leads ultimately to death of the entire organism. So obviously it cannot be explained simply by cellular senescence, just in terms of sensu stricto logical argumentation. So then I say, nonetheless, a surprisingly large number of aging pathologies have been linked directly or indirectly to the senescent process. So I always try to bring you back to what we do know, and then whatever is speculative, I want you to keep that always in mind as well, because this is science. So that means that we're constantly doing new research, and the more we learn about things, the finer the optic gets. And so it's always a movable feast, although we do learn a great deal and understand a great deal during that process of continuing our research endeavors. Okay. All right. So let's proceed. So either normal cells, normal in quotation marks, or young cells, meaning not particularly aged in terms of their cell cycle repertoire, can meet one of two fates. Now, of course, these are generic fates, but let's just go through them. You can have genetic alterations, and these can include mutations or outright deletions of entire introns, for example, or maybe even exons. That all happens in the normal genome, the diploid genome, and that will affect, of course, gene expression and cell function. Those mutations can sometimes lead to a loss of activity or a gain of some activity that could definitely lead down a different pathway for cellular fate and ontogeny. So when that happens, you can generate, even with normal cells, both an aging and a cancer phenotype. So normal cells can start to age, younger cells can start to age. They're not necessarily identical, right? Although their phenotypes may overlap. What happens then down this pathway is what we can say are epigenetic alterations. This has to do with that proliferation of covalent modifications of cytosine residues that are associated in CPG island regions of promoters or enhancers, elements within the DNA acting cis in configuration of activity, because it's all DNA. And those methylations or acetylations of the cytosine residues, sometimes ubiquitinations, and sometimes palmitylation, and even other rarer things like succinylation of cytosine residues, will alter gene expression because of the location and the degree of that covalent modification, likely affecting RNA polymerase binding as well as all the other binding proteins that are necessary for normal transcription. And again, those mutations that were set in place at the early phase of this alteration would then now be altered once again, because epigenetic alterations would lead to a progression down this pathway of aging and cancer phenotype, shown there in that multicolored processing. In other words, multiple mutations and multiple changes in gene expression. Now, the other way of looking at it is that normal and young cells can first have epigenetic alterations. Stress can induce this without causing mutations in the genome. Again, this has to do with covalent modifications of cytosines, and even more so if the epigenetic alterations are occurring first, you get changes in histone modification. Remember, these are the basic proteins which wind up DNA and make up chromatin. So the histones, lysine residues, particularly in histone 3, but in other histones as well, that are cohering basic proteins that make up the nucleosome, those alterations will also change the integrity and the ability of that chromatin to be unwound and therefore expressed in any genetic locus. Okay? So those epigenetic alterations, as I say, change chromatin structure, and that will affect gene expression and then ultimately, of course, cell function. Right? So these are not mutations in the DNA sequence. They're alterations of the structure of the chromatin. Right? That can lead, in some circumstances, to an aging and cancer phenotype. It's initiated. And then on top of that, you can now obtain, because of other stress phenomena or the increases in reactive oxygen or xenobiotics, or simply an aging cell making errors during recombination repair, for example, you can pick up genetic alterations. And now you have, again, a progression of aging and cancer phenotypes. Now, that's not going to lead to the same lineage. So the order in which those changes occur, either genetic or epigenetic, will then yield different downstream modified chromatin, and therefore different cellular fates and tissue fate, and ultimately the fate of the organism. Right? So the changes in morbidity ultimately leading, again, we're all talking about at the end, the mortality of the human. Right? Okay, so that's an important consideration. Now, the senescence response, there are multiple causes. We've talked about this in the past. Persistent telomeric and genomic damage. This has to do with telomere shortening and lack of telomerase activity. Of course, oncogenes can be expressed, which are normally regulatory genes, often in cell cycle. And you can also alter kinase cascades and therefore have strong mitogenic signals, which can cause rampant cellular division. Right? Epigenomic perturbations, again, we already explained what those are, covalent modifications of DNA and protein. Also, I should mention that the production of microRNA is also an epigenomic perturbation. That, of course, is going to change gene expression at the RNA level. You also can have tumor suppressor gene activation. If there are mutations in those tumor suppressors, you, of course, are going to get often cell proliferation or uncontrolled cell division or cell cycle aberrations, right? Such as overproduction of DNA. So you get multi-DNA copies within one cell. Those are common mutations in cells age or as they confer into a tumorigenic fate. Right? And that then will give you the consequences. Tumor suppression can occur if you have tumor suppressor genes which are not altered. You can get tissue repair if DNA repair is working correctly. You can get age-related tumor progression, however, and you can also get age-related apoptosis or some kind of degeneration, usually in the form of programmed cell death. And that's basically, I think, what it says down here. Let's just take a look real quick at what's written in this figure. Causes and consequences of cellular senescence, you see. Cellular senescence in response to potentially oncogenic stimuli. Stimuli include damage of DNA, and then, of course, you need DNA repair. Telomeres elsewhere in the genome, strong mitogenic signals, pretty much what I've just explained to you. And again, the consequences of cellular senescence are myriad. They're essentially irreversible growth arrest, can suppress tumorigenesis. This is normally what happens in younger cell lineages or in cell lineages that are not likely going to be conferred into an oncogenic lineage, right? But also you get phenotypes of senescent cells that can promote optimal tissue repair, which is also good. Senescent cell phenotypes, unfortunately, can also, ironically, and I wouldn't say that without some irony, fuel the development of cancer and thus can cause or promote the degenerative diseases of what are normally found in aging. Again, cancer, and I can just tell you, and you already know this, cardiovascular disease, two major causes of death. And I would say superimposed on that is autoimmune responses, which can also trigger both of those death persuasions of the system, but also can be byproducts of it. Okay. All right, so those nice figures. So we talked about genomic and epigenomic stress. Then it's going to impact the DNA repair pathway. Other types of stress, for example, some kind of xenobiotic, right, or UV light or whatever happens to happen to direct DNA damage. The DNA damage repair then, but if you get persistent damage repair, you're going to turn on the suite of genes, P38, which is a cell cycle orienting gene, protein kinase C, and of course you're going to generate more reactive oxygen. That's going to turn on this P16 Inc4A we've talked about, which is then going to turn on a really important oncogene family called PRB. That's going to generate heterochromatin, and that can actually cause growth arrest. Persistent DDR, though, can also generate P53 activation, which will block the senescence-associated secretory phenotype, which can also be fed in by this pathway because of transcription factors like NF-kappa B and the CEPP beta pathway, which we've talked in past lectures. So with going through P21, you can also get growth arrest, but SASP is not going to be involved in this process if P53 is expressed, right? So this is a regulation of senescence, but also linking it to DNA repair. Therefore, euchromatin versus heterochromatin. Remember, heterochromatin is collapsed chromatin, and when it's collapsed, you get a decrease in gene expression, and that's going to give you this growth arrest. So growth arrest might look good because it doesn't look like cell proliferation, of course, but remember that this is not a healthy phenotype. So this phenotype, unfortunately, can also lead to massive programmed cell death. And then when programmed cell death starts to take off, as we'll see later in this lecture, sometimes that can lead to an oncogenic event and then therefore turn back on the oncogenic pathways, leading, of course, to tumorigenesis. So these pathways can be bifurcated and insulted anywhere in that pathway. And when that occurs, you can switch from programmed cell death to cell proliferation relatively easily. just because of the juxtaposition of all these transcription factors and these kinase cascades regulating cell cycle. Yeah. All right. Now, cellular senescence refers to essentially an irreversible arrest of the cell that occurs when cells experience potentially oncogenic stress. This is supposed to prevent tumorigenesis. So the permanence of the senescence growth arrest enforces the idea that the senescence response probably evolved, at least in part, to suppress the development of cancer earlier in life. This is probably why we see cancer not showing up as frequently in younger populations as in older populations. It has to do with the genetic programming. So the senescence arrest, of course, is considered irreversible because no known physiological stimulus can actually re-stimulate senescent cells to enter the cell cycle. They're at the end of the cell cycle. They're not going to keep on dividing. Mitosis is going to cease, right? But you will generate that senescence phenotype, which is secretory, which can then alter adjacent and associated cellular beds, right? Now, molecular biological manipulations, for example, sequential inactivation of supertumor suppressor genes, can also cause senescent cells, unfortunately, to begin to proliferate. So this is obviously an error in metabolism somewhere, right? Gene expression, metabolism, both. So there may be as yet unrecognized physiological circumstances, these would be pathophysiological, under which the senescence growth arrest does become reversible. So you want that senescent phase to be irreversible, but when it's not, because of all the mutations you can well pick up in an older senescent cell, obviously this is going to lead to a pleiotropic association of potential tumorigenesis. So I say, regardless, the senescence arrest is stringent. It's established and maintained by two major suppressor pathways, which we were just looking at, the P53, P21 canonical pathways, and the P16, INK4A, PRB pathways, which we also have experienced many times in these talks. Now recognized as a formidable barrier to malignant tumorigenesis. That's why you don't see it through most of the age of the organism. There are rare events, and these are, again, because of terrible insults to the system, because of xenobiotics or other kinds of potential carcinogens or heavy-duty stressors, which can then corrupt this whole process earlier in life, right? But we're talking about later in life here. Consistent with the whole view we're describing here, cells undergo a senescence in response to a host of potentially oncogenic stimuli, or perhaps just their sequelae, downstream processing of that event, is what I mean by that. In addition to arrested growth, senescent cells show a widespread, let me move my picture box here, changes in chromatin organization and ultimately gene expression. Now that is, of course, an epigenetic or an epigenomic phenomenon. Those two are different, and I'll talk about that later. All these changes include the secretion of numerous pro-inflammatory cytokines. It's not all one word, sorry about that. Chemokines, which cause cell attractants and mobility of cell lineages. Growth factors, which allow cells to proliferate. Proteases, which allow the extracellular matrix to make a home for potential oncogenic events, such as the metalloproteinases. And a feature termed senescence-associated secretory phenotype we've mentioned many times, which has that really wonderful nickname SASP. I like saying that word SASP. I don't even know why, because I don't particularly like a lot of S's in my words. Now you know. So SASP will then mediate responses in a senescent cell. You're going to get potentially, look at this, angiogenesis, cell proliferation, resistance to chemotherapy if you're already dealing with an oncogenic event. Epithelial to mesenchymal transition, never good because that's leading on the way to hyperplasia, or lack of descriptors so that the epithelia will remain epithelia. You go back to a mesenchymal stage, which means those cells proliferate in multiple lineages. Stem cell renewal, same process, another error, and then differentiation of those stem cells. Inflammatory response, obviously, is coherent with this. And then also you can get tissue repair. So myriad activities of the SASP phenotype. Many factors compose the SASP, have numerous biological activities, all highly dependent upon whatever the physiological context is, such as the cellular bed, right? All those activities include stimulation of those processes we just talked about. So you can have then a many different SAS factors acting simultaneously within and outside a cellular lineage. Okay, so that's what these hexagons are supposed to be in this diagram. One day I won't do that anymore, but I don't know when. All right, so let's now proceed with this discussion in more deep detail. Cellular senescence causes. Cellular senescence first formally described approximately 50, maybe almost 60 years ago now, when Hayflick and colleagues showed that fibroblasts do not proliferate indefinitely in culture. They reached what's called the Hayflick limit. Those cells were said to have a finite replicative lifespan when they're normally dividing. And later they undergo replicative or cellular senescence. Sometimes that's referred to as the Hayflick limit or replicative or cellular aging, the senescent cell phenotype. Number of divisions that cells complete upon reaching the end of their replicative lifespan is called that Hayflick limit after that particular researcher. The link between that limit and the aging was for many years conjectural and tenuous because people kept looking for a direct readout, and what they really should have been looking for are coincidental, consequential effects of that limitation. Hence, the discovery of the SASP, you see. Because you have to realize that once you reach that limit, you're not through with changes in phenotype, to put it another way. So largely on the base of replicatively senescent cells appear to be degenerated, and although they remain viable and metabolically active. Degenerated in what way? They can't hit cell cycle again. Okay, that's what that meant. Now, again, 50, almost 60 years ago, it was evident that most cancer cells do not have that finite limit, and that's why they continue to proliferate. Hence, you pick up mutations that become proliferative, and then you make a real mess out of the cellular bed, right? Because now you've got an oncogenic event, massive cell replication, but you're replicating cells that are already old, which means they've picked up any number of mutations, right? So this is all bad news, but this is what happens as the organism ages. Now, paper published in 2015 in Cancers, that's a journal out of Basel, tells us the following. Cancer development is linked to obesity and infections, which affect cancer incidence. Check this out. About the same. Up to 20% of obesity linked to cancer, and infections can get up to about that as well. Common feature is, of course, inflammation. When acute inflammation is a necessary requisite in protecting the body, of course, against normal infection. Tissue stress and even injury and genomic mutations pleiotropically then result in dysregulated metabolism intracellularly. And all the signaling pathways too are altered. That leads to an inability of the entire organism to return to homeostasis, often promoting chronic inflammation. You can imagine why autoinflammatory diseases get picked up as one ages, you see. Because that means inappropriately recognizing self-antigens and generating an immune response. What kind of immune response? One that is pro-inflammatory. Not a good thing, very common as you age. So inflammation changes the homeostasis, as when hyperglycemia precludes dysmetabolism of carbohydrates and lipid metabolism in particular. And that generates glucose toxicity associated inflammation, such as what happens with insulin resistance in type 2 diabetes associated with obesity, and sometimes metabolic syndrome. And of course, then you get outright tissue damage from that. Chronic inflammation predisposes the body to chronic disease, such as autoinflammation, cardiovascular, and even cancer. And that's ultimately what can lead to, right? That's why cancer is the leading cause of death in humans. Just look at the predisposition of inflammatory bowel disease, if you want an example, and hepatitis B and C infection, when you're talking about the liver, and how both of those map onto, respectively, colon carcinogenesis, and HCC, or hepatocellular carcinoma, UC. And we've covered both of those at great length, particularly hepatocellular carcinoma. I know I've spent, at least 10 hours of lecture on that since last October. Yes, it's been that long talking about aging. It's hard to believe, isn't it? But aging is a long process. Haha. Okay. Group 6A, calcium independent PLA2, phospholipase A2, or IPLA2 beta, other name of the protein, which is an enzyme, cleaves a phospholipid at the SN2 position of the glycerol backbone. These would be glycerol phospholipids. And they generate lysophospholipid and a fatty acid. Generally, that 2 position in many phospholipids, mature phospholipids and membranes, is going to be that omega-6 fatty acid, 20:4, arachidonate. That can be significant disruption of inflammatory equipoise in ways I'm going to describe throughout from this paper we're looking at, 2015 paper. ILPLA2 beta elicits a physiologically important function, including phospholipid remodeling. Obviously, when you remove a fatty acid from the 2 position of the glycerol backbone, you can now use an acyl-CoA to put a new fatty acid in there. For example, one of the omega-3 very long-chain polyunsaturated fatty acids, which is going to change a lot of the activity of the initial inflammatory response because of changes in pro or anti-inflammatory eicosanoids, which of course are oxygenated, roughly 20 carbon chain length fatty acids. Those are called the eicosanoids, right? Such things as leukotrienes from the lipoxygenase pathway, or from the cyclooxygenase pathway, the prostaglandins. Okay? And we've talked about these in great deal because we are all lipid biochemists here because your professor is a lipid biochemist. I'm sure you really well remember all the pathway discussions of thromboxanes and leukotrienes and prostacyclins and outright prostaglandins in our earlier discussions of inflammation. All right, so an inhibition of that enzyme, IPLA2 beta, induces apoptosis. Interesting. Partly by preventing arachidonate incorporation into phospholipids. I'm going to get more into that in a moment, don't worry. That's what the asterisk reminded me of. Whole body deletions, giving you, this is of course in a mouse model, right? Doing a double knockout, where you have IPLA2 beta, double knockout, double negative mice, have been produced. And they do it, of course, by homologous recombination and negative selection. And that shows 2 major phenotypes. With a reduced fertility in male mice, defects in insulin secretion by pancreatic beta cells being the other. Male infertility, or at least a reduction in fertility in the male mouse, as well as defects in insulin secretion come from the pancreatic beta cells. So that's interesting, right? 2 different, what look like disparate phenotypes. Now that's caused, it's believed, by the reduction of arachidonate-induced calcium store in the ER and increased apoptosis because of that in the B cell mitochondria and the beta cell mitochondria, of the pancreas. That's how it knocks out that insulin secretion. Don't you know? The lack of the ability of mutant mice to secrete insulin, therefore, then results in the observed sensitization of glucose intolerance upon feeding with a high-fat diet or treatment with the beta cell toxin, which can induce diabetes, and that's called streptozotocin. Talked about that also a lot in the past. Upon aging, that's getting a little bit old there for the mouse, 1 to 2 years, the double knockout lineage show a loss of bone mass as well, associated with decrease in body weight, and their brains exhibit, whoa, look at this, neuronoaxonal dystrophy. So you're starting to get some phenotypes that look like aging. Hence, IPLA2 beta deficiency, double knockout is a good model for that, at the whole body level can elicit systemic effects associated with chronic inflammation in several tissues and essentially leading to injury, such as seen in the bone and brain of, there you go, the senescing or aged mutant mouse. All right, so you see why this is interesting for our discussion. Okay. Now, here is where that asterisk shows up. It's come from an earlier paper, 2006, in one of my favorite journals, Journal of Lipid Research. So what is this paper referring to? It's about this arachidonic acid release from that phospholipase A2. Now check this out. This is really more explanatory. This is why you listen to me in my Authentic Biochemistry podcast, because I go into the literature and explain what the other paper that we're really dealing with or putting up front and on tap is discussing, so that you get the entire depth of understanding. That's why it's authentic biochemistry. Don't you know? Okay. Well, arachidonic acid participates in a reacylation-deacylation cycle for membrane phospholipids. That's called the Lanz cycle, named after Bill Lanz, who used to work, I think, somewhere in Ann Arbor, Michigan. In fact, my postdoctoral advisor, John Olrogi, I think, got his PhD with Bill Lanz. So that's part of my lineage too, indirectly, of course. So anyways, this Lanz cycle serves to keep the concentration of free fatty acids in cells at a very low level. Now, you know from listening to my multiple lectures that free fatty acids are toxic to the cell. Free fatty acids are toxic to the cell. If they're esterified as oxygen esters or as thioesters, then they're in metabolic play, and they themselves are not, since they're strictly toxic. If they're free fatty acids, they can melt the membrane and cause, again, this toxicity, this fatty acid toxicity or lipotoxicity, and damage membranes. Once you damage membranes, you damage a cell. You can open up an inflammatory bed. Don't you know? Of course you do. Okay. Now, with this paper, to manipulate intracellular arachidonic acid levels in U937 phagocytes, these are cells that have been isolated for the study, they use several pharmacological strategies to interfere with that cycle, the acylation, deacylation, reacylation cycle, for the two position of the glycerol phospholipids in the membrane. Remember, that's the Lanz cycle. They use inhibitors of arachidonic acid reacylation, namely they use thimerosal and triacin C, which block the conversion of arachidonic acid into the CoA thioester so you can reacylate, and also a CoA-independent transacylase inhibitor that blocks the movement of the arachidonic acid within the phospholipid. So you can get transacylases, where you take a fatty acid from one phospholipid and exchange it with a fatty acid in the two position of another phospholipid, and you do an exchange, a fatty acyl exchange, right? That's how those two inhibitors function. You can see how they would disrupt the Lanz cycle, right? All right, now they go on. In addition, they used overexpressing group 6A phospholipase A2. Overexpressing, which means you're going to get a lot of phospholipase A2 activity, which means you're going to get a lot of deacylation of the glycerol phospholipid within the membrane, okay, if it's active enough. So they overexpress it, and that's what they're getting here. And you have, therefore, they're saying that this enzyme has key roles in controlling Basel fatty acid deacylation. Of course it does, because it's a phospholipase. And they're talking about doing this in these phagocytic cells, which is their research study system. All these different strategies result in expected increases, of course, in cellular-free arachidonic acid. All three of those would do that, but also in the induction, now check this out, of programmed cell death by, of course, apoptosis. Moreover, they say, when used in combination with any of those aforementioned drugs, right, which further enhance the amount of free arachidonic acid, AA itself was able to induce apoptosis at doses as low as 10 micromolar. And of course, it would. But there's two reasons. One, it melts the membranes because you have a free fatty acid. And number two, it has a lot of double bonds, and you know those double bonds are going to get oxidized, which is going to generate in situ within the membrane, chain reaction, reactive oxygen species, and further proliferation of radicals within that double membrane, triple membrane, quadruple membrane system. That's going to lead to membrane disruption big time, okay? Writ large, as some say. All right. Moreover, when used in combination with any of the aforementioned drugs, as I just said, okay, blocking cyclooxygenase or lipoxygenase, that's going to take the arachidonic acid and make all those oxyicosanoids, oxy containing molecular oxygen, that is, had no effect on the induction of apoptosis by A. So it had nothing to do with the downstream processing. It has to do with the lysophospholipid in the membrane and all that activity of the free arachidonic acid. Don't you know? See, because if they made the acyl-CoAs or the transacylases were making new, fully born glycerophospholipids with two-position filled, you know you're going to be able to do a lot of icosanoid production, right? As long as you induce it. They're saying it's not what was going on here, right? So they're differentiating two different potential, sometimes pleiotropic, working together, modifying to generate a high stress response. They separate the two and say, no, in this case, it's not the icosanoid production, which naturally it does involve a lot of oxidative metabolism. So essentially what they're saying here, arachidonic acid levels within the cells may provide an important cellular signal for the onset of apoptosis and the perturbations of the mechanisms controlling arachidonic acid reacylation with that two position of the glycerol backbone, and hence free arachidonic acid availability may decisively affect cell survival in the form of programmed cell death, which is what they saw, which is what they're talking about. So that's why I went back to this paper, you see. This paper reveals how the work in 2015, 9 years later, was resolved. This is how research works. Previous papers, you build on those previous papers, you generate a hypothetical deduction, you then design experiments, do the experiments, you generate data, you analyze that data, and if the data is analyzable and is solid, has low standard error, then you can start to derive evidence from that and then authenticate that evidence. And once you do that, then you can ultimately lead to an induction and move on to yet another maybe hypothesis, right? Or to begin to develop some kind of theory of the process. This is how it's done, by using the literature. So here we go. Back to the paper, 2015 paper. The livers of the double knockout mice showed increased apoptosis associated with increased inflammation. Makes sense. While the double knockout mice physically appeared to be normal, they exhibited in vivo susceptibility towards LPS-induced liver injury, CON-A, that's concanavalin A, induced autoimmune hepatitis, and the dextrane sodium sulfate-induced colitis. Okay? IPLA2 beta mediates, of course, apoptotic cell clearance through the generation of this lysophosphatidylcholine, which is the other product, thus regulating the speed and directionality of the monocytes during chemotaxis. Okay, this is really important to keep in mind. That's normally what it's doing, right? Because it's removing that fatty acid, and that fatty acid is going on down to its ultimate fate, right? So this is going to redirect how monocytes function to generate then their full-blown pro-inflammatory response. It's all very linear and logical, right? So this defect in apoptotic cell clearance, right, which is going to be a real problem to the tissue bed, might explain the observed susceptibility of the double knockout mice towards stress-induced injury, which is going to be the apoptotic mark, right? Damaged and dying cells promote seek and destroy signals. Seek and destroy signals. This is going to be the innate immune response coming in, where a seeking signal is, of course, the the lysophosphatidylcholine, right? Because you missed that fatty acid, which is coincidental with the cleaved, once you make that LPC, that lysolipid, it's coincidental with the cleaved caspase-3 mediated apoptosis, which is coming again from the mitochondria, which is how this whole thing was put together, as I talked to in the introduction. Okay. So mice deficient in the seeking signal, lysophosphatidylcholine, for example, fail to remove the apoptotic cell, because they're deficient in that signaling, because they're deficient in the phospholipase 2 beta, you see. So they're not making the lysophosphatidyl-choline. Got it? Okay, good. So they fail to remove apoptotic cells, which become secondary NUC product once the apoptosis starts to kick in. And that subsequently triggers a pro-inflammatory response by the immune cells, because then you get necrotosis, which means the cellular contents are released, and now you get a full-blown pro-inflammatory response, which could, even include non-acquired response from the T cell lineage, T cell receptors, working in tandem with antigen-presenting cells now, right? Then B cell activation on you go to make antibodies to. So mice lacking G protein coupled receptor 132, which is also known as G2A receptor, or R, which is indeed the lysophosphatidyl-choline receptor, yes, indeed, they develop an autoimmune disease that looks a lot like lupus. Now you start to see how lupus plays such a dramatic, significant role here, which is what I say all the time anyway. But now you just get a good taste of it by looking at this paper. Again, this paper is about cancer in the aging. We're looking at a mouse model, but you see that this works very well because we know all of these processes, albeit differentially regulated, also occur in humans. Okay, so I want to make that clear of why we're launching into this. Now, so the results of the paper ultimately said this: a deficiency in this phospholipase A2, specifically the inducible PLA2 beta, increases apoptosis in spleen and primes splenocytes for ultimately, didn't I tell you T cell response? Your Th1, Th17, boy, that's a gangbuster pro-inflammatory response, right? Those are the CD4 lineage T helper cells. So IPLA2 beta deficiency increases apoptosis in liver associated with suppressed cytokine release by the Kupffer cells, which are, of course, immune cells in the liver. Sublethal dose of CD95 Basel treatment primes the mutant with the double knockout, Kupffer cells, for an enhanced M1, that's a pro-inflammatory, macrophage, cytokine release. Okay? So sublethal dose CD95 Basel treatment primes the mutant liver lymphocytes, which is where we're at now. We're talking about now, because of this M1 cytokine release, now we're starting to talk about the Th1, Th17 activation because of antigen presenting to those T cell lineage, to the TCR, right? And also to the CD3, the co-adapter, right, to fully turn on those T cells. And so you get a Th1 cytokine release. That's the response, right? And Th17 reduces Th17 cytokines. Okay. Now with aging, an increase in apoptosis in these MLNs, these mutant liver lymphocytes, right, was associated with an inherent loss of B cell cellularity and decreased lymphoma. So you see, you get this degradation pathway, but you don't generate the oncogenic pathway. So you see where this is an important fulcrum or focus point between either going to cell proliferation on one hand, or which would be oncogenic and tumorigenic, or cell degradative cell death, which can also, of course, cause tremendous damage, such as liver necrosis, right? Which is going to cause morbidity in that organism as well. And later on, could actually trigger down the road, because of all this hyperinflammatory response, sure, a proliferative response, and you could still get hepatocellular carcinoma. You see how that works? It's a timing, it's a temporal mediated response that has to do with these differential damaging systems to the tissue related to the aging process because of membrane lipid changes, because of alterations in, for example, in this case, phospholipase activity, generating potential signals for pro-inflammatory responses, or directing that lineage away from one system, the oxygenated fatty acid system, using that arachidonic-CoA to make the eicosanoids, but going right into an apoptotic necrototic pathway, generating the inflammatory response. See? So there are two distinct pathways we're discussing here. Yeah. But they will lead to the same problem down the road to the organism. So I'm going to stop here because then we're going to get back into genomic damage. I wanted to cover that cancer paper, which I think we did pretty effectively. So let's go to here, end the show. And I think we're ready to say that we're finished with today's lecture. I want to remind everyone that we're in a drive right now to get people to subscribe to the YouTube lectures, and not only subscribe, but to donate to our fund. Now, you can go on my Authentic Biochemistry podcast site, and there's a direct place for you to donate directly to the PayPal. Okay, so I want you to keep that in mind because we want to be able to produce a lot of these video lectures because I know people like them, and I like giving them because we can really show you how science is done directly, authentic biochemical research science. But I also like doing the audio ones. The audio ones we can go a little bit faster. We can cover a little bit more ground. We tend to do more review article papers so we can cover whole segments of a system. But I also like talking about these individual papers. And here, the best way of doing it is by doing these video lectures. So please donate by going to our PayPal site and donating as vigorously as you can, and subscribe to my channel because that means I can do this more often because it's take time away from other things I'm doing. And if I'm deterred from that and allowed to do more of this, I can get to a larger audience and we can have more and more people learning more and more authentic biochemistry. Okay, so that's my pitch. I don't do it often enough, and the reason I don't do it often enough is I'm not a very good salesman. Okay, but I think I'm a pretty good professor and research scientist, and that's what I'm trying to do with you. But at the same time, everybody's got to be fed, and so we need to get an income from this. And that's what I'm pitching right now. I know you dig what I'm saying and you understand. This is that kind of world. So this is Dr. Dan Guerra saying on the 21st, the first day of June, 2021, bye for now.